Today I'm going to go over protein synthesis in case you missed the notes for protein synthesis. And what protein synthesis is when your DNA makes a sequence of proteins that allow your body to work. And protein synthesis has 2 steps, and they are transcription and translation. I'm going to go ahead and go through the different steps and draw them. So we're going to do transcription first. And transcription occurs in the nucleus. And it's when you are converting DNA into mRNA. And in this case, the M stands for messenger. I'm going to draw transcription on the next page. So we're in our nucleus. And we have our DNA. And it's in our double helix. And DNA is going to split in half. So we're just going to have one side of the DNA, one strand. And this strand is going to have 9 sides to it, or 9 rungs to it. So then we're going to use this DNA strand, and I'm just going to put some random bases down there. So T, A, G, C, T, A, C, G, T. And mine didn't really line up very well, but you have one base per line. So we're going to use this DNA to find our mRNA. So you need to know the base pairing rules that go from DNA to mRNA. So DNA to mRNA. Your bases for DNA are A, T, G, G, and C. A will go to U, T will go to A, G will go to C, and C will go to G. You'll notice that RNA does not have T's, so that's why we're going to be using the U. So now we need to find the corresponding strand of mRNA that matches up with the DNA that we have. So T will go to A will go to U, G will go to C will go to G, T will go to A will go to U, C will go to G, G will go to C, and T will go to A. So there's our mRNA strand, and that's transcription. Now, the mRNA is going to leave the nucleus and go to the ribosome, which then becomes translation. And translation occurs in the ribosomes. And in translation, you are changing, or rather using your mRNA strand to make amino acids, which are building blocks for protein. So we have to use our original strand from our mRNA. And I'm going to lay it down on the bottom like this. So 1, 2, 3, 4, 5, 6, 7, 8, 9. And let's see, it was AUC. GAU. And then GCA. Alright, so when you get into your ribosomes, because we're dealing with translation, this is translation still. The first thing you need to do is break your mRNA strand up into groups of 3 bases. So here's one, and here's one. So we have 3 of them. Each one of these things is called a codon. And a codon is just a group of 3 bases. So the mRNA strand is going to code for proteins, which are made up of amino acids. So we're going to be finding amino acids that correspond with the codons of the mRNA. And the amino acids are attached to a thing called tRNA. And tRNA is transfer RNA. So it's transferring the proteins or the amino acids to the mRNA. So we need to go ahead and draw some transfer RNA, and they kind of just look like this. So this is our tRNA. And they are also going to have 3 letters that go with the mRNA codons. So these mRNA codons match up with the tRNA. And when you're doing mRNA to tRNA, the base pairing rules are like this. A and U, and then G and C. Okay, so now we need to find our tRNA. So these little doodads, like the spaceships, are tRNA. So A will go to U, U will go to A, and C will go to G. G will go to C, A will go to U, U will go to A, G will go to C, C will go to G, and A will go to U. These green things that are attached to the tRNA are called anticodons. I kind of ran out of room there. So now, once we have our anticodons and our tRNA, we have to find the amino acids that go with this information. And how you do that is you're going to use an amino acid chart or a codon chart. And we have one on page 211 in our textbook. And I'm also going to end up giving you a chart or something like that later on, so you can use that as well. And I'm going to go ahead and erase a little bit because we have to write some stuff on the top of our tRNA. And what we're going to be writing on the top of our tRNA are the amino acids that we found from the chart. We're still going to be using this information right here, the mRNA, to find the information on the chart. The tRNA is just used to transfer the proteins or the amino acids to the mRNA strand. So we need to find AUC on our chart. So AUC, and AUC is isoleucine. So I'm just going to draw a box. So isoleucine. Then I need to find the next one. And that's GAU. GAU is aspartic acid. And the last one is GCA. So let me erase a little bit more right here. Put a little box. And GCA is I'm looking for it. Alanine. And that's how you do protein synthesis. Again, after you watch this video, let me know if you have any questions because it is a little confusing.